Welcome back to part 2 of chapter 10. In the first part of chapter 10, we talked about DNA replication. For when cells wanted to divide, they would have to do DNA replication before they could do that. And then we talked about the central dogma of biology, taking DNA and reading it to make RNA, and taking that RNA to make proteins. So transcription is the process of taking DNA and making RNA, and then translation is the process of taking that RNA and making proteins. Just like transcriptions, translation has 3 parts: initiation, elongation, and termination. So in initiation, what happens is, remember, translation occurs, proteins are made at the ribosome. So this blue part here is part of the ribosome. That's the small subunit. And to start, you have methionine, the first amino acid, bind to the mRNA and come into the small unit of the ribosome. Now we're ready to get started. So what happens next is the large subunit comes in, and we've got our whole setup ready to go. You've got 2 different parts here for where the tRNA, the transfer RNA, can come in to bring the amino acids. The first site is the P site, and then you've got the A site. They come into the A, and then they shift over to the P. And we'll look at that during elongation. So during elongation, you've got the codon recognition. So that means the codon here matches up with the anticodon on the tRNA. 3 nucleotides in a row. The next 3 will code for the next amino acid. And the first one is always AUG. It always codes for methionine that we saw in initiation. And then during elongation, we're going to keep adding more in. So what happens is you make peptide bonds. So remember, the bonds between amino acids are peptide bonds. And once that's made, the first tRNA is released, and this next one shifts over. And we keep moving down the mRNA, adding on more amino acids. So here you can see we've got a 4 amino acid long polypeptide. We bring in the next tRNA that matches the anticodon to the codon. We form the peptide bond next. And then once we've done that, they're attached together. That first tRNA is released, the second moves over. And now we're ready to bring in the next corresponding tRNA. What happens to this tRNA is it's going to go out and find the specific amino acid that binds here so it can do its job again. And then last, termination. So we keep moving on, adding on more amino acids until we reach the stop codon. There are a couple different codons that code to stop. And after that, we've got our primary structure of a protein. And what happens next is it's going to become the secondary structure, tertiary, and then it can be combined with other proteins to make its quaternary structure. So let's look at an overall review. I think it's a really good idea to do. So this is a beautiful picture. DNA to RNA to proteins. So DNA, you can go through replication, make new copies of DNA, and then you could also do transcription, where we open up the DNA, we make RNA. And this happens in the nucleus if you're in a eukaryotic cell that has a nucleus. So there, the DNA never leaves the nucleus except when the nuclear membrane dissolves during mitosis, only during cell division. Otherwise, we make the RNA, we process it, it leaves the nucleus, finds a ribosome, either free-floating or attached to the endoplasmic reticulum, and we build proteins. We combine amino acids together to make these polypeptides, these long strings that, when added together, make proteins. So here's a nice picture review as well. We've got our DNA, we've opened it up. We've got the enzyme RNA polymerase coming through, reading the DNA and attaching the matching up RNA. Then we've got that RNA and we process it. We cut out the introns, we only excite, read the exons, we add a cap and tail to it. That leaves the nucleus, goes to the ribosome. We have the initiation once methionine combines onto the mRNA at the ribosome. Then you've got elongation. You keep binding the tRNA to the mRNA, adding on the amino acids, shifting over until you've got a finished product. You find the stop codon, you have termination. So in eukaryotes, as I said, you've got a nucleus, transcription occurs in here, translation occurs in the cytoplasm at the ribosomes. In prokaryotic cells, there is no differentiation, it all occurs into the same space. Okay. So transcription and translation are the processes where we take DNA, we make RNA into proteins. Those proteins can be for cell structure, they can do enzymes, they determine our cell function. Now let's look at mutations. A mutation is any sort of change in the nucleotide sequence, any changes in the nitrogenous base pairs. So the example we've got here is the sickle cell anemia that we've looked at in previous chapters. We have just one change of amino acids that changes our codon. So now instead of coding for glutamic acid, we're coding for valine, different amino acids with different structures and different properties that change the shape of hemoglobin. There are two types of mutations. The one that we looked at with sickle cell, and is shown here, is a base substitution. We've changed a single nitrogenous base. Sometimes these can be silent, they don't change the amino acid sequence. Sometimes they do. Insertions and deletions are really bad mutations. If you take one of those three U's and you take it out, now you have this whole frame shift. So instead of going 3, 3, 3 and looking at the amino acids it codes for, now what we're doing is going 3, 3, 3 down here. And you've got different amino acids that are going to be coded for all the way down. Generally, that will cause the protein to be non-functional. So mutations can arise from errors in DNA replication. Every time we make a new set of DNA, when we do cell division, we have errors. Our DNA polymerase catches almost all of them. It only misses 1 in 100 million or so. Like viruses, they don't have DNA polymerase checking it. So what they have is more like a rate of 1 in 100,000 base pairs are wrong. So we've got these checks in place for DNA replication, but we've also got chemical and physical agents, these mutagens, that increase the rate of mutations. So this is a really nice picture here showing radiation from the sun and X-rays, chemicals like cigarette smoke, and then infectious agents, they can increase the rates of mutagens as well. I think this is a really interesting example. Back in the 1950s or so, there was a drug for sale that helped pregnant women with their nausea. It's called thalidomide. Many of the babies born from women who took thalidomide had mutations in their development that caused them to have improperly formed limbs. So the mutations that we looked at have generally been harmful, but they're so important. They are responsible for genetic diversity. And where most mutations are harmful, every rare once in a while you'll have genetic mutations that are selected for that give the organism an advantage and have it pass on more of its genetics to offspring. And it makes the world an interesting place to be. Last topic I wanted to discuss briefly is viruses. viruses are really cool. If you ask a virologist if a virus is alive or not, they'll say, I don't know. They can do everything that we talked about in the first chapter, but they can't do it without a host. So viruses, they have genetic material, they have packaging, and that's pretty much it. And what they do is they go and they infect host cells, and they cause those cells to become virus factories. They have either DNA or RNA, they only have one, and they go into the host cell, they insert it, and then that host starts making the viral proteins. What's really neat is that the RNA viruses, some are called retroviruses. What that means is that you take the RNA and you make it back into DNA before you go through DNA RNA protein. And that backward step, there are no checks to make sure we're not making mutations. So viruses like HIV that are retroviruses mutate at much higher rates than even normal viruses. And then the infected cells generally produce viruses until the viruses exit the cell and the cell is killed. And depending on the type of cell, like in our bodies, we have some really cool responses to viruses. Like our T cells, these are some of our white blood cells that do immune functions. When they get infected with a virus, they start making the viral proteins, but they also make these proteins called interferons. And interferons go out and warn other cells. They say, hey, it's too late for my cell, but they go to the next cell and they warn them. The cell puts up a block so that the virus can't get in. And that's all we have for Chapter 10. I will recommend a video for you to watch to understand translation and transcription better. The video that I generally show in class is this from RNA to protein synthesis. It does a really nice job of visually showing you the steps of translation because it can be quite confusing. There are a lot of really good videos out there on these topics, and if you need more help, I would recommend looking there as well as in your homework. Have a great day!